I am a PhD student in the Integrated Coastal Sciences program at East Carolina University. I also am working for the state of North Carolina Office of State Archaeology as a contractor for some heritage at risk work with hurricanes from Hurricane Irma and Hurricane Michael. But this is talking about my dissertation project. So this is a proposal defense. And as I just said, this is the first time I'm doing it. My defense is in 20 days. So you get the first go at this. So I hope you enjoy it. There will be no results. This is just a proposed project on wooden shipwreck stability in a shallow water tidal fresh ecosystem. So we'll be moving through these 4 topics in general, background on the project, kind of where this came from, the interdisciplinary framework that I'll be using for this project, the methodology, and then why is this important in the grand scheme of things beyond cultural heritage, as we all know, as that's why we're here today. So a little bit of the background. This image is actually the type of wreck or the ship I'll be working on. It is a emergency fleet vessel. It's a wooden ship that was built during World War I to essentially build a bridge to Europe. So when the US entered the war, they didn't really have a shipbuilding program. So they launched this huge shipbuilding program to build vessels of all types. So concrete, iron, steel, and wood. And this is what the wooden vessels ended up looking like upon completion. So remember that as we move forward, what this kind of looked like. So within the background research realm and what we already know, we have spent years studying how sites are affected underwater. But a lot of these have been within their own realm. So physical forces, we know that we've done numerous studies where we've seen how currents and wave action can move ships or shipwrecks and break shipwrecks up. They can move artifacts. We know sediment movement can cover and uncover sites, exposing them to different factors. On a chemical level, we know there are chemical interactions that are happening, particularly for iron and steel. So as iron corrodes underwater, it forms a concrete-like material around it, encasing it that can offer a level of protection, but it can also offer a level of degradation. And from a biological side, we know that these shipwrecks end up being like coral reefs. So they function in a reef-like setting. Creatures live on them. Different corals inhabit them, but this can also happen at a smaller level that we can't necessarily see. So for wooden wrecks in particular, shipworm is a big problem in warmer, salty waters, which is where those worms typically live, and they'll work their way into the wood and eat away at the wood. We also know that microbes are there, but we're not sure in what state what those microorganisms are and really how they affect the wood. So that leads us to what aquatic microbes are. So they can be any small microscopic organism that's floating around in the water, that's living in the sediments in the water, that's living in conjunction with the other organisms in the water. So coral reefs have a symbiotic relationship with microorganisms. And individually, these organisms are in fact impacted by their local conditions. So think water temperature, pH, dissolved oxygen will all dictate the types of organisms that are found in a certain place and their ability to function as an individual, but that also impacts the community structure. So thinking of community structure, it's how many different types of species are living in a community. And we can see that, in a forest with different birds and trees and insects and deer and stuff like that. But the same thing happens at a microscopic scale, that we have different like bacteria and archaea and eukaryotic microorganisms living in the same community. And in the case of what we're going to sort of talk about today, they live in a biofilm. So they they kind of latch together and form like this slime essentially on a structure that binds it together, and collectively they function as this
I bet she's going to catch up with us in a second.
community to survive and thrive. So you provide resources for them. And so most of these studies in aquatic microorganisms have come from marine and estuarine environments. We know very little about what's happening in freshwater environments. So taking that a step further and looking at aquatic microorganisms living on submerged cultural heritage, in wood in particular, we do know a little bit. We know they're there. We have had several studies that have shown that erosional bacteria and other organisms are living on wood. In Florida, you could just walk out to the pier and see like algae growing on pillars, and that's essentially a microorganism community. And so several studies have looked at succession of these organisms on different types of woodfalls in different depths of an environment, going from anywhere from like 100 meters to like 2,000 meters. So that doesn't necessarily help a lot of us help us really in understanding what's happening on shipwrecks, because a lot of our shipwrecks tend to also be in shallow waters. But they have shown that there are similar structures to that successional stages and some continuity between how this community is forming. But there's also been a few studies that have looked at how the wreck microbiome, so what the wreck took down to the bottom of the ocean with it, and then joined on by organisms already living there, how that has impacted the environment in general. So these shipwrecks, particularly in deep marine waters, are there by themselves, right? So they form this island. And there have been several studies coming out of the Gulf of Mexico following the Deepwater Horizon incident that were particularly looking at how the hydrocarbons impacted these communities. But they were able to do some studies to understand how that wreck microbiome is impacting the surrounding sediment. And what they've seen is that as you move away from the wreck, that impact lessens, which is kind of cool to understand. What is that impact of a wreck on an environment? We think a lot about how the environment is impacting the wreck, but if you want to think about it from another angle, the wreck is also impacting the environment, and it's important to understand how, if we're going to manage all of these things collectively. So a step further into just general shipwreck and submerged cultural heritage preservation, for wood, most of this started with ex-situ understanding. So they would recover artifacts that were wooden and try to understand what was happening to them in order to preserve them, right? So think the Vasa, the Mary Rose, these wrecks were brought up and they were like, okay, we know that water has impregnated them. How did this happen so that we can stabilize them? With a 2001 UNESCO convention, in situ preservation was really pushed, and this started this drive amongst archaeologists to figure out how to preserve wrecks underwater, which required them to understand some of these more dynamic relationships to preserve a wooden wreck underwater. Now, a lot of what we think is that for the most part, they'll be pretty stable by themselves. They can be left alone. But we're not really sure if that's the case, because a lot of this has happened at a singular time frame, or they'll go out 5 years in the future and look at it again. But how are those things changing, and what is causing those changes? So what we have in the gaps of all of this is that we know that wood can be fine, but we also know that wood can degrade. And so with what the studies we've had, this project hopes to kind of fill that gap and quantify that rate of degradation for a wooden wreck, and understand how all of these different factors are working together. So how are microorganisms and the water and just the archaeological history in general pulling together to create a state of preservation or a state of degradation? And from a natural science side, there's a lot we still don't know about microorganisms and how they kind of, so what biogeography here is how they connect within space and time. So how are they spread out across the wreck, and how does that community structure change over time? And so from the natural science side, this project is hoping to provide some insights into not only wood biogeography, but in a fresh, shallow water environment, which is very much lacking in terms of our understanding from a microbial side. So the project framework is trying to bring all this together. And in doing so and thinking through this process, I wanted to kind of like, okay, we have our thought, our wooden shipwreck preservation. What are the different factors that impact that? And we talked just a little bit about it with the natural side, so our biology and our hydrology. So we know that different organisms use it. We know that the effects of the environment, so the water chemistry, so our pH, our dissolved oxygen, our temperature, all influence that, the movement of the water, the movement of the sediment. But from the other side, there are a lot of questions about how did the systemic culture and how did the post-depositional culture like impact that as well. So in thinking about this, these are all of the red that you're seeing are the things that this project's hoping to address. Obviously, this is a huge question, so we'll see how far we get. But we can also think about this from the other angle, from the natural science side with the microbiome in the middle. And what's important here is this differentiation for biologists and natural scientists tend to think of our shipwrecks and as just a substrate. But with that substrate comes a whole other realm of possibility coming in. And, there's been a lot of work on understanding how shipwrecks are degrading, particularly iron and steel, with the hydrocarbons and the release of gas and, oil into the environment that could cause this huge natural disaster. But that requires us to think about that structure, or biologists to think about that structure from a different view, that it's not just a substrate for organisms to live on, that it holds its own importance. So this project is asking this question: how long and to what extent do a wooden shipwreck's construction, historical depositional condition, archaeological state, microbial communities, and environmental conditions influence and contribute to its stability? So really what it's proposing is that a rate of change can be quantified by bringing all of these different influences together, and individual changes in these communities and conditions, so seasonal shifts in temperature, seasonal shifts in water quality, seasonal shifts in community structure will be evident and visible and contribute to that rate of change. And with that, because this is such a large question and there's so many components to it, there are a set of disciplinary questions for each discipline that this is bringing in. So archaeological is looking more at that systematic and post-depositional context, as well as the density of the wood itself. The microbial ecological questions are delving into the biogeography of that microbial community. So how is it changing across time and across space? And then our hydrological questions are there to kind of place this in a larger context of what is happening in the environment and with climate change. We know things are changing, so how can we spot that change happening? So to think about this in a theoretical sense, this is going to rely very heavily on the behavioral archaeology. And so this is kind of the flow model I came up with based on Schiffer's work for a traditional terrestrial site, but putting it in an underwater context and bringing in that outside influence of climate change. We can see that up until the archaeological record, it's following our pattern of things are built, because decisions are made that could contribute to its decay or stability or life in general. And once it's just deposed, it becomes an archaeological record. And then all of these different forces are at play. And with that creates a new record, and this just becomes a cycle, right? So things will change within our, influences that'll change the site, which will then start us back again. So this is just a continual process, but we don't know how long that process takes and what is happening throughout that process. So methodologically, we have a bunch happening from all of these different disciplines to pull in, archaeological frameworks and microbial frameworks and hydrological frameworks. So this is relying on a case study site that's on the eastern seaboard in the Potomac River in Mallows Bay National Marine Sanctuary. And so within this bay, there are about 100 wrecks, all dating to the EFC vessel or EFC era during World War I and after World War I, when they were deposited for salvage. There are a few other wrecks in there, like this one that dates to the 1970s. But all of these are the same build. So it's this really neat environment to do this work in, because it's hoped that this wreck as an archetype can kind of provide some management guidelines for all of these wrecks within this bay and the larger sanctuary, and the methodologies can then be applied elsewhere. So this is our specific wreck. It's about 80 meters long and 20 meters wide. It's called Iowa. It was built in New York in 1918. It never saw action during the war because the war ended shortly after. It was sold to a salvage company and dragged in the Potomac River in the 1920s. And that is where it's been sitting ever since. So this project will start with some desk-based research to understand long-term environmental conditions. So we'll be looking at satellite imagery and buoy data from the larger Potomac River all the way down to the Chesapeake Bay to kind of reconstruct what's been happening over the last couple decades, and see if we can see those changes. We know things are changing. Information has been coming out of the Chesapeake region saying things are changing. So this is an attempt to quantify those changes and understand what's been happening. We'll be also doing some historical research to get into the details of the wreck itself and the whole era of these wrecks and how they were built, what was happening, what wood was used, how were they constructed, and that construction will feed into the development of a three-dimensional model that will be done in Rhino 3D to reconstruct the vessel as it was intended to be. So these all had specific designs that were coming out of DC, and part of what we're trying to understand is, were those decisions that DC was saying, hey, you should build a vessel like this, actually being implemented in these shipyards as this process was happening super fast once we entered the war. So we'll also be doing a lot of field work. So we'll be doing archaeological field work in the bay to completely map the wreck. So we'll use traditional baseline offset mapping. We're going to map it to a 1 to 25 scale, meter scale. We will do photogrammetric recording. We're taking a lot of photos across the site of key features that can help us identify potential changes from the construction plans. And then we'll be using a resistor graph to measure the timber density, which I will come back to later. So hold on to that thought. We will, every time we go into the field, we will be doing hydrological testing. So we'll bring a YSI, which is a water quality monitoring device, and it essentially drops in the water and it gives you readings for the different sensors you have on it. So we will be measuring pH, temperature, salinity, and dissolved oxygen, all factors that are known to influence not only microorganisms, but also the wreck itself. And finally, we'll be doing microbial work to understand the biofilm and the related community. So the microbial fieldwork is a big component of this, because this is what we will be doing seasonally for the next 2 years. So we'll be scraping the wreck biofilm And then we'll be taking sediment samples and water samples and filtering out the organisms and extracting their DNA to see who's living in there and what that can tell us. So the wreck biofilm scrapings will be happening at 6 different locations around the wreck, on the interior side and the exterior side of the structure, to see if there's a change between those two sides, knowing that some of this water inside of the wreck can't get out some of the time. So this is a tidal environment. So there's times where the wreck is completely submerged, and there's times when the top of the wreck, as here, is exposed. So there's pockets of water up here that may never leave the wreck, but we're not sure if that has any influence on the microbial community. So we're trying to assess that. And with that, we'll have 3 sites that will undergo replication to ensure that what we are seeing is correct. And these are all happening on frame stations. So you'll have, so what we're sampling is the outer hull and the inner hull planking at a frame station. So there's a frame right here. So the rib of a ship and the planking on either side. So from the environmental side, for our microbial sampling, we're doing sediment sampling that will take off in 2 transects, one from the bow and one from the stern. These are happening at regular intervals for the first 5. So there's one here at 0, which is our stern, and then 25, 50, 75, 100. And then we're going out to 200. And this methodology and distance came from the work out of the Gulf of Mexico with Layla Hamden and the Deepwater Horizon incident. Granted, she was doing it on a much larger scale out to 2,000 meters. We don't have that ability. We're kind of bound by the bay here. And she was also doing cross transects. So going this direction. We've decided not to go in that direction because of the quantity of wrecks that are there that would probably, have similar microbiomes, so we wouldn't necessarily see what we're trying to see. Does microdiversity related to the wreck decay over time? And if we're going into another wreck, we're not necessarily going to see that because it's going to have its own microbiome. So we're also taking 3 water samples from the bow, midship area, and stern. And those will all be filtered in replicate to see who's living in the water and how that wreck structure is influencing the surrounding water, what's coming off, what's not coming off. So with that, obviously that's a lot of data we're gathering. And to kind of answer all of our disciplinary questions, we'll be looking at everything from a disciplinary start to begin with. So with our archaeological questions and understanding construction, differences and behavioral decisions made back in the 19 teens, we'll be overlaying all of our data with that Rhino model to see what changes are evident, if there are any evident changes, but we'll also be using it to kind of quantify how much of the wreck has disappeared since it was first deposited there. Now we know there was some salvage, so how much can we contribute to them stripping and burning the wreck to get down to this level, or was there other influences at play? From a microbial side, we will be doing a lot of statistical analyses to understand the differences in community structure, both at each site and each sampling period, and bringing that all together to understand changes across the wreck structure and across the environment at each sampling period. And hydrologically, we'll be charting that same sort of time trend. How are things changing in the bay? How are things changing from bow to stern? Can we suss out a difference at all? So to give you kind of a little guide here to what the site actually looks like within the bay. So up here at the top, we have the construction plan that was made by the US government and said, this is what you're building. In the middle here, we have a scale drawing of the site as it is on the bay floor. And then this is our photogrammetric model. And you can see they line up pretty well in terms. So this breakwater is present and evident in all of them. And so this just is giving you a little sense of how this process is going to work from an archaeological comparative standpoint. But the big part of our archaeological analysis is coming with our timber density. And so as I mentioned earlier, we'll be using a resistograph to gather that density data. So this is me with the resistograph in the pool. We tried to get pictures on site and it didn't work because the water was too dark. But this is what it looks like. So it's this pneumatic drill. So these are air hoses filtering down, and it's sending a needle into the wood. And so as that needle is pushing against the wood, there's friction being exerted on that needle. And so it's measuring how much friction and how much force is needed to get that needle to drill into the wood. And with that comes this curve. These two different curves, our feed curve and our resistance curve, to see what the stability is of the wood, but it can also count tree rings. So all of these ups and downs are the varying tree rings within this timber. And this gap right here is actually a change between two pieces of wood on the shipwreck. So this is our outer hull planking, or no, our ceiling planking, and then this is the start of our frame. And so what we're doing, we're measuring resistance and density at each of those microbial sampling locations. So all of these 6 blue dots here in the middle, or squares in the middle, align with where we were sampling for our microbes. So we're tying them together to see if that community is influencing the density of the wood. We're going to do additional resistograph sampling in a few other places, including the bow and the stern, both the top and the bottom, to kind of understand how things in general, irregardless of the larger question of how these factors influence each other, but just in general, what is the overall stability of the wreck from top to bottom in this environment. And so to bring this all together, we're going to do several different components here. So we have statistical analysis, visual analysis, and then spatial analysis. So statistically, our response variable and what we're trying to get at is that wood density, right? But all of these factors influence each other. So we'll first start with looking at our microbial community as a response variable and placing it against all of the environmental factors. And an analysis similar to this, so this is a coordination analysis, which basically says how is diversity changing with the influence of, say, salinity or water temperature. So we're trying to understand what is impacting this community's diversity, and is it changing seasonally? Is it changing annually? Is fall always the same? And so once we understand those relationships, then we're going to attempt to start to understand those relationships with timber density. And so we'll be doing several analyses like this. So this is just a linear regression, but looking at how that diversity is impacting timber density. And from that density data, we can extract quantifiable numbers and use them to create and analyze these relations statistically that will hopefully contribute to our understanding of degradation over time. Visually, we will again be looking at the wreck as it is compared to what it was supposed to be, but from these different data sets, we can visualize them individually as well. So with our microbial data and our resistance data, we can interpolate that data across space to see how things are kind of changing in terms of like diversity. Is diversity waning in certain areas? Is diversity growing in certain areas? Is resistance, is it more stable on one end versus another? So these can be used to kind of see that and put some of that statistical information that not everybody, might like to understand, but in a more visual way to see what's happening. And with that, we're going to attempt to do something kind of cool that kind of expands on general spatial interpolation and put these data sets into a machine learning software in Arc Pro to construct and predict more accurately those changes over space and potentially over time. And so essentially what's happening with this Geo AI software in Arc Pro is we'll take our variables from a measuring or from a sampling cycle, drop them in to the program, tell them how they relate, and it will conduct a regression analysis. And with that analysis, we can then train a model to hopefully predict the degradation and higher quality and higher accuracy across that surface. So this is doing that, but our Geo AI model will hopefully give us a finer resolution than these, large spaces that are saying it's just kind of degrading or, it's getting more stable, but what's actually happening in here where we didn't measure at a finer scale. And so, as I said, this is over a multi-year period. So hopefully we're going to start sampling this winter. If not, we'll start in the spring. But we're doing 8 sampling cycles that will measure microbial sampling and water quality. Over those 8 samples, we will go back next summer or fall with the help of the Maritime Studies program here at ECU for a field school to do a final resistograph sampling and archaeological analysis again, see if anything large has shifted. And that field work will all be promoted and supported by the Maryland Historical Trust. The rhino modeling is currently happening right now and will happen throughout the spring with support of the national sanctuaries or the National Marine Sanctuaries and the National Marine Sanctuary Foundation with some congressionally earmarked funding. And the hope is that this will all be done by the spring of 2026. Analysis and all. So as you've seen, we have some data already, and a lot of this was all collected with the help of the Program in Maritime Studies annual summer field school in 2022. And this is just the amount of data all of those guys helped me collect. I couldn't have done it without them. That's where all the pretty pictures have come from. That's where the site plan has come from. And with that, we've been able to create this foundational data set to base this whole project off of. So we took rec sample or biofilm samples and microbial samples, and that is giving us a starting point to understand what is there, because we don't know what's there to begin with. So this is helping us fill that hole to then start these seasonal samplings. Okay, so you might now be asking why should we care about this and what is this importance? So from an intellectual side, this project is filling several gaps in both the social science side and the natural science side. This is one of the first attempts that I've been able to find that's actually trying to quantify a rate of degradation. And within that, it's using behavioral and site formation theoretical concepts to help answer that question. We think about some of those behavioral decisions in its own context and site formation in its own context. So this is trying to bring them together to be like, they do influence each other. From a natural science side, this is filling a lot of gaps in our understanding of both wood microbiology and fresh and tidal fresh aquatic microbiology. There's a lot of unknowns that still exist in those realms because the microbiological world is so huge. So this is helping answer some of those questions. These are some novel approaches. The resistograph itself, there's only about 10 out there in the entire world. Most of them are in Europe, and most of them are being used to measure telephone line poles, and in the underwater context, piling. So there's a few in Amsterdam that are being used to ensure the integrity of the pilings to support the city. So this has never been used in an archaeological context underwater. There have been a few studies from architectural studies that have used this resistograph in a way like this, but never to really just understand a site for the site's sake. And GeoAI is still relatively new, and in terms of archaeology has never been used in a context like this to understand and predict changes over space and time. And from a microbiology standpoint, there have been a few studies that have utilized machine learning, but never in sort of this joint context to bring it all together. In terms of the broader impacts, hopefully this will be used to managers and as a methodology for understanding degradation, a way to prioritize sites or use data and the understanding that we gather here to kind of prioritize sites. We'll be doing local outreach in the community. I'm setting up talks with the local archaeological society in Charles County, Maryland, to talk about this project. And just expanding the local knowledge, not only of the history and the archaeology of Mallows Bay and these sites, but also bringing it together with the other side and the natural side and making it this whole grouped thing. And then students, as I said, the Maritime program has been instrumental in gathering this data and will be instrumental in gathering this data in the future, but hopefully bringing some biology students along and just helping people understand that these disciplines in different departments don't need to be separate from one another, and they really should be together. So overall significance, this is bringing together from a cultural side, it's constructing and evaluating these relationships through a different lens that may have been used underwater. We use site formation underwater all the time to understand how the site is being impacted, but it's not, again, bringing that systemic context and necessarily bringing the systemic context and the depositional context together to understand how that is overall influencing our structure. It's, again, hopefully going to create a degradation rate that is bringing these different factors together and is creating a methodological framework for assessing these factors in the future. From a natural science side, it is expanding on the concept of biogeography, which is relatively new in microbiological studies, are relatively untested in microbiological studies, and bringing that into this wooden framework that really is understudied. And a lot of these microbial studies happen at one place at one time. They don't have the funding and the ability to easily go back and do these things seasonally and over a number of years. So in doing so, we're hoping to show some potential trends that may be useful for understanding the microbiome in general. And overall, this is a new ability to understand these forces in relation to new things going underwater. And they may not necessarily just be cultural resources, but infrastructure that may be wooden or have large wooden components as climate changes are going to enter this new environment. And this gives us hopefully a more detailed understanding of how those factors are created and what's happening within the larger context. And this is, an integrated methodology that's bringing it all together and crossing those barriers, because they really shouldn't be separate because they exist together. So yeah, that's all I have for you.